Next one is enzyme inhibition. So as we are saying that enzymes have the catalytic property or catalytic ability, that catalytic ability is inhibited because of some other substances. So those substances which are inhibiting the activity of the enzymes are called as inhibitors, and the process or mechanism where the activity is inhibited is called as enzyme inhibition. So there are three types of enzyme inhibitions are there. Those are called as competitive inhibitors or competitive inhibition. Second one, non-competitive inhibition. Third one is a feedback inhibition. So let us start with the first one, the competitive inhibition. In competitive inhibition, some substances will inhibit the catalytic activity of the enzyme, and these substances are called as inhibitors. So these inhibitors are structurally similar to substrate molecule. As they are structurally similar to the substrate molecule, so when the inhibitors reach the active sites of the enzyme molecule, now the enzyme consider that as a substrate molecule and it will provide the active sites for that inhibitor. So once the inhibitor will come and occupy the active sites, now the enzyme is not able to provide the active sites for the actual substrate molecule. That is the way how this inhibition is taking place. So this type of inhibition is called as a competitive inhibition, and the competitive inhibition can be reduced when more number of substrate molecules are present. So let us take one of the example here, that is there is an enzyme called as succinic dehydrogenase enzyme. So this succinic dehydrogenase is specific to the enzyme substrate called as succinate or succinic acid. So this is the enzyme, this is the substrate molecule, and malonic acid is a competitive inhibitor for succinate molecule. Let us see this in the form of a diagrammatic representation, where this is an enzyme molecule and this is active site present on the enzyme molecule. Here this is a structure of the inhibitor, that is a malonic acid, and this is a structure of the actual substrate, that is succinate molecule. Then here, before the substrate come and occupy the active site, the inhibitor will come and occupy the active site. Now once this inhibitor come and occupy the active site on the enzyme molecule, now what happens is the substrate will be remain non-reactive. That means it can't participate, or it cannot occupy the space active site on the enzyme molecule. So this type of inhibitors are called as a competitive inhibitors. So some of the competitive inhibitors are useful for treating the cancer and also some bacterial pathogens. So let us take the second one, the non-competitive inhibition. So here also the inhibitors are present, but these inhibitors are not structurally similar to the substrate molecules. And one more thing, that these inhibitors occupy other sites on enzyme other than active site. That is a difference, because in the competitive inhibition, the inhibitors are structurally similar to the substrate molecule and they are occupying the active site only, and making non-availability of the active site to the substrate molecule. But here these non-competitive inhibitors are structurally not similar to the substrate molecule, and at the same time they will not occupy the active site, but they will occupy any other site on the enzyme molecule, and the effect of that process will be on the active site. That is, there will be a change in the shape and the structure of the active site. Now this active site is not being reactive, or it will not fit into the substrate cannot fit into the active site because of the change in the shape and structure of the active site. So let us take here, this is a enzyme molecule with this is the active site, and this is actual substrate molecule, where the structure of the substrate is exactly similar to that of the active site, and at the same time the non-competitive inhibitor. Before the substrate come and fit into this active site, the non-competitive inhibitor will fit into some other space or some other place. So what happens is the nature, actual shape of that active site will be changed. So here the inhibitor is not attaching to the active site but to the other part. So because of that reaction, what is happening, the shape of that active site have been changed. Now as the shape of the active site have been changed, it is not available, or it is not exactly fitting to the structure of the substrate molecule. So this type of inhibitors are called as a non-competitive inhibitors. So some of the examples of non-competitive inhibitors are salts of heavy metals and cyanides. Third type is a feedback inhibition. Let us take one example related to feedback inhibition, where glucose molecule will react with the ATP molecule to form ADP and glucose 6-phosphate. So this reaction is catalyzed by an enzyme called as a hexokinase enzyme. Here in this process, this is one of the reaction related to the glycolysis of respiration, where the accumulation of this glucose 6-phosphate will show effect on the hexokinase enzyme. So this is what the feedback inhibition on the hexokinase. That means the hexokinase will become inactivated. Once it got inactivated, it cannot participate in the further reactions where the glucose had to be converted into the glucose 6-phosphate molecule. So likewise, the accumulation of excess products will inhibit the enzyme activity, and these are otherwise called as allosteric modulators also. There are some other type of inhibitors are there, those are called as allosteric inhibitors. So this concept of allosteric inhibitors was given by Jacob and Monod. According to the scientist, some substances are the inhibitors which will bind to some other sites, that is allo means or we can say allosteric sites on the enzyme. So actually the name have been derived based on that activity only, in which allos means other, steric means site, or steros means site. So the inhibitor will bind to other site on the enzyme, that is other than the active site, and the effect of that allosteric site attachment of the inhibitor will show the effect on the reaction. So the activity, catalytic activity of the reaction, catalytic activity of the enzyme will be reduced. This type of inhibitors are called as allosteric inhibitors.